Welcome back, everyone. So we're going to switch gears and look at cell division in our basically our germ cells, which basically, instead of producing genetically identical cells, the job of meiosis is to produce our haploid gamete cells. So that's what we're going to learn about in 6.6, meiosis. Okay. So now that we have an understanding of sexual reproduction, we need to be able to produce those gametes to basically half our chromosome count, so that our gametes have half as many chromosomes, so that upon fertilization we have the correct number of chromosomes in our offspring. So again, because we are diploid organisms, we need to produce haploid cells that can fuse together to form diploid offspring during fertilization. So that whole process to produce these haploid cells is called meiosis. And meiosis is basically just a series of events that, similar to mitosis, arrange those chromosomes so that they separate into the daughter cells correctly. There are some similarities to mitosis in meiosis. So there is an interphase, just like in mitosis, where each chromosome is duplicated. However, instead of just one round of nuclear division, like in mitosis, meiosis actually has 2 rounds of nuclear division. So that actually results in 4 haploid daughter cells. So by the end of this video, you should be able to describe the behavior of chromosomes during meiosis. You should also be able to describe the cellular events during meiosis, as well as explain the differences between meiosis and mitosis, and explain the mechanisms within meiosis that generate genetic variation among the products of meiosis. So just to review, we already know that sexual reproduction requires fertilization. So basically we are taking the DNA of 2 separate genetic sources, right? So it's the union of 2 cells. We have the sperm from one cell, and the genetic information from a sperm cell, the genetic information from the egg cell, uniting to form a zygote. Okay, so we have our diploid zygote. And so each one of these gametes, these sex cells, must contain 1 set of chromosomes. Okay, so in us humans, we are diploid. And so most of our cells in our body are what we call somatic cells. So somatic cells are non-reproductive cells. They contain, basically the nuclei of each one of our somatic cells contain 2 copies of each of our chromosomes. And we talked about those copies being homologous. One of those copies came from our mom, one of them came from our dad. So meiosis is a process where we start out with a diploid cell, and that diploid cell actually comes from a specialized cell called a germ cell. So this is different from a somatic cell. This is a germ cell, which will undergo meiosis instead of mitosis. And that germ cell will end up producing 4 haploid gametes. So each one of those gametes, instead of having 2 sets of chromosomes, will have 1 set of chromosomes. Okay. So we're going to do just an overview of meiosis first, then we'll watch a video of it, and then we'll get into the details. Okay. So the overview of meiosis includes interphase, and then meiosis part 1, and then meiosis part 2. So interphase of meiosis is the same as mitosis. So we'll see that there's, interphase is just the same. We have G1, that stands for gap 1, which stands for growth, and then the S stage is where the DNA is replicated, and each chromosome has a identical copy held together with a centromere. And then we have the G2 phase again. So the first part is exactly the same as in mitosis. Then we enter the first phase, nuclear division, meiosis part 1. So in meiosis part 1, it's quite different from mitosis, okay? And the reason being is that we have the homologous chromosomes come into play here. Now remember, homologous chromosomes, you have two copies. You inherited one from mom and one from dad, okay? And what's unique about meiosis is that the two copies hang out together during meiosis, okay? Particularly meiosis part 1. So in meiosis part 1, the two copies in prophase part 1 and prometaphase part 1, they are really close together. And even in metaphase part 1, they are next to each other, okay? So the first part of meiosis part 1, the goal is to separate these homologous chromosomes from each other, okay? So we end up separating them. And so they end up in separate daughter cells. So the goal of meiosis part 1 is to take our diploid cell and make it haploid. So basically separating the homologous chromosomes into separate cells so that each daughter cell just has one set of chromosomes. So we went from 2 sets of chromosomes to 1 set. So now each daughter cell is diploid at the end of meiosis part 1, or I should say at the end of cytokinesis. Okay, so we have our 2 daughter cells, each have 1 set of chromosomes. But notice the sister chromatids are still attached at this phase. So that's why we have to go through meiosis part 2. So meiosis part 2 is actually the same as mitosis. So the job is just to separate the sister chromatids from each other. Okay, so that's why we have to have that second round in order to separate the sister chromatids from each other. Okay, so it might seem confusing at first, but don't worry, we'll go through it step by step, beginning with an amoeba sisters video. So let's watch that.
Have you ever wondered how 2 siblings can have the same mom and dad and still look so different? Well, today we're going to talk about a process that makes that possible, a process called meiosis, not to be confused with mitosis, which sounds unfortunately similar. Mitosis makes identical body cells, like your skin cells and stomach cells. Recall from our mitosis clip that since it makes identical body cells, mitosis is important for growth and for repair of damage, or to replace worn-out cells. But not meiosis. Meiosis is a process that contributes to genetic variety. Meiosis also doesn't make body cells. It makes sperm and egg cells, otherwise known as gametes, the fancier word. You might recall that humans have 46 chromosomes. That's how many chromosomes most body cells in your body have. But there are some human cells that don't have 46 chromosomes. Human sperm cells and egg cells have 23 chromosomes. Why the number difference? Well, if a sperm cell has 23 chromosomes and an egg cell has 23 chromosomes, when they come together, that makes 46 chromosomes. That will allow a newly formed fertilized egg to develop into a human. Meiosis is what we call a reduction division because you have a starting cell that has 46 chromosomes, and your ending cell, the sperm and egg cells, have only 23 chromosomes. Before we can start getting into the stages of meiosis to make gametes, we have to remember what happens before meiosis can even start. Actually, this also happens before mitosis. It's the stage known as interphase. And if you remember interphase, it's when the cell is growing, it's replicating its DNA, it's carrying out cell processes, and just like mitosis, interphase happens before meiosis is even going to start. So the starting cell has 46 chromosomes, and you have to duplicate those chromosomes in interphase before meiosis starts. That basically means you are duplicating your DNA, since chromosomes are made of DNA and protein. Ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, because the sister chromatids are still attached, and we are actually counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture. But you went from 46 to 92 chromatids. Little tricky there. We have a detailed video that explains these chromosome numbers before and after replicating in interphase that can be useful for meiosis. Okay, so interphase checklist done. Now we can move into meiosis. You might remember the mitosis stages PMAT. The P was for prophase, the M was for metaphase, the A for anaphase, and the T for telophase. And the good news is that in meiosis, you still use those terms. But because meiosis is actually a reduction division, you're going from 46 chromosomes to 23, which means you actually divide twice. So instead of mitosis, where you divide one time and do PMAT one time, in meiosis you're going to divide twice, and therefore do PMAT twice. And because of this, in meiosis, you put numbers after the phases to indicate whether you're in the first division or the second division. So let's dive right in. So let's start with the very first step, prophase 1. One thing I like to remember about prophase is pro, this actually means before, and it kind of helps you remember that it comes before all the other stages start. This is where the chromosomes are going to condense and thicken. They are actually going to line up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. They're going to match up. It is during this prophase 1 that this amazing process occurs called crossing over. Now I know crossing over probably sounds like something very different, but this is actually a really awesome process because this is when the chromosomes, they're lined up in homologous pairs and they have this way that they can transfer their genetic information and exchange it between each other. It's kind of like these chromosomes flop over each other and they do a little genetic information exchange here. It makes for what we call recombinant chromosomes, which can eventually contribute to the variety that we were mentioning that siblings can have even when they have the same parents. More about that later. Now we move into metaphase 1. In metaphase 1, think of the M as standing for middle. The chromosomes are now going to be in the middle of the cell. It's a little bit different though from mitosis because these chromosomes are going to be in pairs in the middle of the cell. So it's not a single file line, they're in pairs in the middle. Now during anaphase 1, think of A for away because the chromosomes are going to be pulled away by the spindle fibers. Then we end with telophase 1 where you have two newly formed nuclei and it becomes obvious that you're going to end meiosis 1 with two new cells. Cytokinesis follows with splitting the cytoplasm, but we're not done yet. On to meiosis 2. The very first step in meiosis 2 is prophase 2. It's not going to be nearly as eventful as it was in prophase 1 though, because they're not going to have homologous pairs of chromosomes. They also are not going to have this amazing process called crossing over. That doesn't happen again in prophase 2. You have your chromosomes, and the spindles are starting to form like they did in prophase 1, but prophase 2 is just not as eventful as having that process of crossing over. In metaphase 2, remember, think M for middle, the chromosomes are going to line up in the middle. This time though, they are in a single file line. They are not in pairs like they were in metaphase 1. Anaphase 2, remember A for away, but this time it's the chromatids that are going to be pulled away by the spindle fibers. In telophase 2, you can see the nuclei reforming, and you can also see that the two cells have divided. There's going to be 4 cells forming. Cytokinesis will follow to completely split the cytoplasm. Now, keep in mind that meiosis in males produces sperm cells, and in females it produces egg cells. Because of independent assortment and also crossing over, you're going to have variety. For example, in a male, the 4 sperm cells that are produced each time, they are all different from each other. They're also different from the starting cell because the starting cell had 46 chromosomes, and the ending cells only had 23. So they are not identical to the original, and they are not identical to each other. This is going to lead to variety, a reason why 2 siblings with the same parents can look different from each other. They still developed from a unique egg and a unique sperm cell that came together. One last thing to think about. Scientists are often looking into the process of meiosis because sometimes the chromosomes don't separate correctly. It's called non-disjunction, when a cell can receive too many or too few chromosomes in the separation. This contributes to some genetic disorders, which is something scientists continue to study. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so that was a good introduction into this whole process of meiosis. And you'll notice again that, some of it has similarities to mitosis, but there are quite a few differences. So let's go ahead and look at the details. So starting off, interphase should just be review for you because it's exactly the same as in mitosis. So the cell goes through the 3 phases of interphase, just like in mitosis. We have the G1 phase, remember, starts with cell growth. And then we have the S phase, where the DNA of the chromosomes are replicated. And so during that phase, we have the sister chromatids forming with the centromeres holding them together. And then we have the G2 phase, where the cell is preparing for its final preparations for meiosis. And then this phase right here, instead of mitotic phase, think meiotic phase. Okay, so now we're going to get into meiosis part 1. So remember, the goal of meiosis part 1 is to go from diploid to haploid. So the goal is to basically take the homologous chromosomes and separate them from each other, so that each daughter cell should have 1 copy of every chromosome instead of two copies. So prophase part 1 starts out very similarly to mitosis prophase. So the chromosomes start to condense just like in prophase. The nuclear envelope breaks down just like in prophase. The centrosomes start moving to opposite poles just like in prophase. The mitotic spindle starts to form just like in prophase. However, there is a big difference here. Now, as the chromosomes are condensing, the homologous chromosomes start to get close together and they pair up. And when they pair up into what's called a synapsis. And during this pairing up into a synapsis, they get so close together that the ends of the chromosomes kind of flop over each other. And when they flop over each other, we can have something called crossing over take place. And during this period of time of crossing over, a little piece of one chromosome can break off and attach to the other, and vice versa. And you can see here that if we have, this is our homologous chromosome, but it's a non-sister pair, okay? So it's not part of, these are not identical to each other. They're homologous, they carry the same genes, but maybe not necessarily the same versions of the genes. So they might break off and attach, okay? And that way, we have these little segments of different chromosomes kind of exchange genetic material with each other. And so the point where they actually exchange genetic information is called a chiasma, okay? So a chiasma is like the plural, chiasmata is the singular for that. Okay, so that's just the structure that forms at that point where they kind of flip and exchange information. And so when they're grouped together like this, you count 1, 2, 3, 4 kind of total chromatids. So that's called a tetrad. So a tetrad is formed from the 2 duplicated homologous chromosomes bound together by the chiasmata during the prophase part 1. Okay, so you end up with this, they're kind of held together at this spot right there. Now this can happen at multiple locations. It can happen up here, it can happen over here. These two can kind of switch too. So it doesn't have to happen at just one location, this crossing over event. And when it's done, we can have little segments of different DNA on each of the sister chromatids. And so when we have that, it's called recombinant sister chromatids. Okay, so recombinant just means like reshuffling of the genes. Okay, so this right here, this sister chromatid is considered a recombinant chromosome. This sister chromatid here is considered a recombinant chromosome because it's got a reshuffling of the genes from another chromosome. This one right here is all blue, so it's non-recombinant. It's original. This one over here is all red. This is non-recombinant. It's original. Okay? So think recombinant is reshuffling of the genes. All right? So that's prophase part 1. We have this main event of crossing over. And you can see that this is one way that we can have genetic variation take place, because now we have some shuffling of the genes. And so because of this recombinant chromosomes, this chromosome is now different from this one. This chromosome is now different from this one. And these 4 chromatids are all different from each other. So when they end up in separate gametes, they're now unique. Okay? Next, we go into prometaphase, part 1. Okay, so prometaphase, part 1, that occurs just like in mitosis. Remember the centrosomes, they start producing that mitotic spindle. So here is prophase part 1, where the nuclear envelope is dissolving. So in prometaphase part 1, the nuclear envelope has fully dissolved. The mitotic spindle has extended and has attached to the kinetochore proteins at the centromeres, just like in mitosis. Okay, so nothing real different about prometaphase part 1 as in mitosis. The only real difference is that the homologous chromosomes are still hanging out together. Okay. In metaphase part 1, the spindle fiber is pushing and pulling the chromosomes until they're lined up in the middle. The only real difference here between metaphase part 1 and metaphase in mitosis is that they're aligned by their homologous pairs. Okay, so you can see them lined up as a tetrad, okay, as their homologous pairs. They're not lined up just, as a bunch of X's down the middle. You see them as side-by-side X's, all right, as homologous pairs. And the way that they're lining up, it's completely random. So you can see how there's a red X on one side, there's a blue X on the other here. Here you have a blue X on one side, you have a red X on the other. So this is what we call an independent assortment of chromosomes. This is another way that we get variation in our gametes because of this random assortment. So in this case, we only have 2 pairs of chromosomes showing. As humans, we have 23 pairs. So imagine all the randomness that can occur of all of our chromosomes as they're lining up in the middle. 23 pairs randomly assorting themselves, that can lead to all sorts of different variation of how they can assort themselves, leading to random assortment in our gametes. Okay. In fact, there's so much assortment that can take place that if we did the math, 2 to the 23rd power, so there's, we could have mom's chromosome one side, dad's chromosome on the other side, or vice versa. So there's, 2 chances, 2 times, 23 possibilities or possible chromosomes. There's over 8 million possible combinations of our chromosome arrangement. Okay, so 8 million possible combinations. So that adds to variations within our gametes. Okay, so that's the second source of variation in our offspring. So the first was the crossing over effect, creating recombinant chromosomes. Now the second one is this independent assortment of chromosomes during metaphase part 1. Okay, So you can see here an example. We have chromosome A. We have, I don't know, big A on one side, little a on one side, big B on one side, little b on the other side. So all the little capital letters on one side, lowercase letters are on another side. So this is one possible way that the chromosomes could have oriented themselves. Here's the other possibility. So big A, little b, little a, big B. And so based on these possibilities, here's an example of the resulting gametes from this combination. Here's the resulting gametes based on this combination. So this is only for 2 chromosomes. Well, we have more than that, right? We have 23 possibilities. So there's 8 million, or we have 23 chromosome pairs. So we have 8 million possible combinations of our chromosomes. All right, so that adds to variation. So in anaphase part 1, we have, instead of the sister chromatids separating like in mitosis, the homologous chromosomes are separating from each other. Okay, so the centromeres are staying together, and it's just the homologous chromosomes are being pulled to opposite poles. Okay, so there they're going to opposite poles. The sister chromatids stay together. Right? And finally, in telophase part 1, we end up with basically the kind of a some cells start having like a nuclear envelope reforming. Some cells start seeing the chromosomes decondense, but not all cells, because we're not quite done yet. But we do see cytokinesis take place. So in cytokinesis, we have the cleavage furrow forming for us, animal cells. And then we have the separation, and we have the two daughter cells forming. Now at the end of meiosis part 1, we have 2 daughter cells, each with 1 set of chromosomes. Okay, so at this point they're haploid. They started out diploid, they ended up haploid. But we have the sister chromatids still attached. All right, so now we are done with meiosis part 1. We've taken our diploid cell and we've made 2 haploid cells from it. But we still have the sister chromatids attached, so that's why we need to undergo meiosis part 2. Okay? So just to review, meiosis part 1, that's the first round of meiotic cell division. And that is also known as the reduction division, because we're taking our diploid cell and we're producing haploid daughter cells. Okay. The next stage is meiosis part 2, so that's the second round, and that's when we are separating the sister chromatids from each other, and that will result in our 4 unique haploid cells. Okay. Now, in between the 2 phases, we have something called interkinesis. So interkinesis is basically just a period of rest that takes place in some species. So during this period, you just had the haploid cells, they just finished cytokinesis. Sometimes there are nuclear envelopes that have formed and you have 2 nuclei present. Sometimes the chromosomes have decondensed a bit. So they're just kind of resting a little bit before they start meiosis part 2. This is not interphase. We are not going through the cell cycle. We're not going through G1, S, G2, okay? Because we don't want more DNA, right? So no DNA replication is taking place. So think interkinesis, not interphase, right? So once it's, had some time to rest, ready to go, then it's going to enter meiosis part 2. So meiosis part 2 is a lot easier to remember because it's basically just mitosis. Okay. So meiosis part 1 is tricky because you got the homologous chromosomes hanging out together. Now we have the homologous chromosomes are separated from each other. So now we enter prophase part 2. So prophase part 2, if we still have that nuclear envelope intact, then it's going to start to dissolve. Okay. The centrosomes start moving to opposite poles. The mitotic spindle starts to form, right? The chromosomes start to condense, just like in mitosis. In prometaphase part 2, the mitotic spindle starts to elongate and attach to the kinetochores at the centromeres of the sister chromatids, just like in prometaphase in mitosis. All right. In metaphase part 2, the sister chromatids align at the equator of the cell, the metaphase plate, just like in mitosis. Okay. In anaphase 2, the centromeres dissolve and the sister chromatids are pulled away from each other to opposite poles, just like in mitosis. And then finally we have telophase part 2, where it's basically the opposite of prophase part 2. So we have the chromosomes are at opposite poles. The nuclear envelope starts to form around the DNA, the chromosomes. The centrosomes start to dissolve. The mitotic spindle starts to dissolve, and the chromosomes start to decondense. And then we end up with cytokinesis, cell division. Okay, the cytoplasm divides. All right, so we end up with our haploid cells. Now, we started out with a diploid cell that has 2 sets of chromosomes. We ended up with a haploid cell, 4 haploid cells that have 1 set of chromosomes. And the haploid cells, if you notice, are each different from each other. So you can see here we have a blue with a little red. This one's all blue. This one's red with a little blue. This one's all red. Okay, so they're all genetically unique. So it's a unique combination of the original DNA. So that is it from meiosis. Okay. So let's do some comparisons. Okay, so first off, let's compare meiosis part 1 with meiosis part 2. So in meiosis part 1, we have in prometaphase part 1, remember how the microtubules, or excuse me, the mitotic spindle, or you could say microtubules, but the mitotic spindle is going to attach to the kinetochores of the sister chromatids. Okay, so you can see where it's attached. It's attached right at the edge. Okay. But in metaphase part 2, the microtubules are attached to the individual kinetochores of the sister chromatids. Okay, so the microtubules are attached to the fused kinetochores, so the kinetochores are kind of fused together. These kinetochores are separate in prometaphase part 2. So there's one difference between meiosis part 1 and meiosis part 2. And so in meiosis part 1, this holds the homologous chromosomes together. Okay. In meiosis part 2, we don't want them held together. Okay. And then so that's one big difference. Okay, so in meiosis part 1, we want the chromosomes, the sister chromatids, to stay together when they're separating in anaphase part 1. In meiosis part 2, the homologous chromosomes have already separated. We want the sister chromatids to separate. Okay, so there's a big difference there. All right. So let's take a closer look at the difference between meiosis and mitosis. So if we remember from meiosis part 1 and meiosis part 2, meiosis part 1 is very different from mitosis. Because the goal of meiosis part 1 is just to separate the homologous chromosomes from each other. Okay, and we also have like that. The homologous chromosomes are exchanging genetic information from each other. They're orienting themselves together during middle metaphase, so it's quite different. However, meiosis part 2 is the same as mitosis, right? We have basically the same thing happening. We have the sister chromatids separating just like in mitosis. So you can see kind of what's going on, the process. Interphase is the same in meiosis as in mitosis. Okay, DNA replication occurs during the S stage of the cell cycle. Okay, we have, let's see, the only, yeah, so basically that's basically the same. Let's see, I'm just kind of seeing meiosis part 1, we take our diploid cell and we make haploid cells from it. So now we have 2 haploid cells. So they undergo basically meiosis part 2, where we end up with 4 haploid cells. Mitosis, we start out with a diploid cell, we end up with 2 diploid cells that are identical to the original. Okay, so that's another big difference. So we're going to watch an Amoeba Sisters video that goes through a comparison of mitosis versus meiosis.
In biology, there are often vocabulary terms that sound pretty similar. Chromosome, chromatid, chromatin, transcription, translation, mitosis, meiosis. You probably have encountered this. When I was first learning about mitosis and meiosis, I learned them both separately first. And then I tried to figure out what was the same about them, what was different, why did they both matter. I would try to compare the stages by flipping through images. You know what would have really helped me? A side-by-side comparison. And that's what this video is. We assume you already have a background of mitosis and meiosis. If not, take a look at our videos on them. But this video is a side-by-side comparison, presented in a split screen. Mitosis on the left, meiosis on the right. Both of these processes, along with the cytokinesis that follows them to split the cytoplasm, are involved in making new cells. Mitosis results in body cells. Meiosis results in sperm and egg cells, otherwise known as the fancy term gametes. Before we start mitosis and meiosis, let's look at what you start with. Your starting cell in both mitosis and meiosis is diploid, written here as 2N. That means it has 2 sets of chromosomes. In humans, that's including 1 set of 23 chromosomes from mom and 1 set of 23 chromosomes from dad. 46 chromosomes total. During interphase, the cell duplicates the chromosomes. When you duplicate 46 chromosomes, you still say there are 46 chromosomes, as the newly duplicated portion is still attached at the centromere region. But there are actually 92 chromatids. Interphase isn't part of mitosis or meiosis, but it's a really important phase because it duplicates chromosomes before we get started. Just to point out, it's really hard to draw 46 chromosomes. We're going to use 6 chromosomes in our diagrams when we illustrate what's happening, because it's much easier to draw and visualize. Oh, and just a fun fact, some insects have 6 chromosomes, like mosquitoes. Unfortunately, I'm not a fan of mosquitoes, but mosquitoes do both mitosis and meiosis. When learning the stages, we get the acronym PMAT, which is helpful for understanding the stages. Both mitosis and meiosis go through these stages, but meiosis goes through them twice, and therefore has a number next to each PMAT stage. We're going to show some basic events for each PMAT stage, but please know that there is way more detail to explore than what we can include in this quick video. Prophase in mitosis. Remember that pro can mean before, and this stage comes before the others. The chromosomes are visible if we say they're condensing, which means they are thickening. Prophase 1 in meiosis. Happening here too, but the chromosomes are actually going to match up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. With each pair, one came from mom and one came from dad. In this formation, chromosomes can transfer their genetic information and exchange it between each other. It's called crossing over. It can make for what we call recombinant chromosomes. Metaphase in mitosis. The nuclear envelope which had surrounded the nucleus was already disassembled before metaphase even started. For metaphase, I like to remember the M for middle, because in this stage, the chromosomes line up in the middle of the cell in a single file line. Metaphase 1 in meiosis. The chromosomes are in the middle as well, but they're still going to be in pairs in the middle of the cell, so it's not a single file line. Anaphase in mitosis. I like to think as the A for away, the chromatids are pulled away by the work of the spindles. They are moving to opposite sides of the cell. Anaphase 1 in meiosis. Same thing, but in this case, it's the chromosomes, not chromatids, being pulled away to opposite sides of the cell. Telophase in mitosis and telophase 1 in meiosis. The chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make two new cells. And they are starting to surround the chromosomes on both sides. This will eventually form two cells. Cytokinesis follows to split the cytoplasm to complete the actual dividing of the cell. So at the end of mitosis and cytokinesis, we end with two identical diploid cells. In humans, they would both have 46 chromosomes. This is great for organism growth. Growing requires making more cells, after all, or replacing damaged cells. On to meiosis 2. Prophase 2. Chromosomes are condensing in both cells. It's not going to be as eventful as it was in prophase 1 because they're not going to have homologous pairs and crossing over. Metaphase 2. M for middle, but this time the chromosomes are in a single file line, similar to how metaphase looked in mitosis. Anaphase 2. Think A for away. This time, though, it's actually the chromatids that are getting pulled away. Telophase 2. Chromosomes are at the complete opposite ends, and new nuclei are forming on each side to make these new cells. Cytokinesis will follow meiosis 2 to completely split the cytoplasm. We are now finished with meiosis, and we end with 4 non-identical cells. Gametes. Males make sperm cells in meiosis, and females make egg cells in meiosis. These gametes are haploid, meaning they have half the number of chromosomes as the original starting cell. In the case of humans, the resulting cells would each have 23 chromosomes. By the way, when a sperm and egg cell combine, it results into a diploid cell, a fertilized egg, otherwise known as a zygote, which will then start a series of divisions using mitosis to give rise to a brand new organism. Well, that's it for the Muta Sisters, and remind you to stay curious.
Alright, so that was a good review of both mitosis and meiosis. So let's see if we can answer some review questions. So meiosis produces blank daughter cells. Is it A, 2 haploid daughter cells, B, 2 diploid daughter cells, C, 4 haploid daughter cells, or D, 4 diploid daughter cells? If you said 4 haploid daughter cells, I would agree with you. So remember, at the end of meiosis, we have 4 genetically unique combinations of the chromosomes, so 4 haploid daughter cells. Here's another question. At which stage of meiosis are sister chromatids separated from each other? Is it during prophase part 1, prophase part 2, anaphase part 1, or in anaphase part 2? So when do the sister chromatids separate from each other? If you said in anaphase part 2, I would agree with you. So remember, in anaphase part 1, only the homologous chromosomes are separating from each other. So anaphase part 2 is when the sister chromatids actually separate from one another. Okay. The part of meiosis that is similar to mitosis is it A, meiosis part 1, B, anaphase part 1, C, meiosis part 2, or D, interkinesis? So what part of meiosis is similar to mitosis? If you said meiosis part 2, I would agree with you. Okay, so meiosis part 2 is basically the same as mitosis. The only real difference is that you're dealing with a haploid cell instead of a diploid cell. Okay, so C is the correct answer. And finally, if a muscle cell has a typical organism, so this is not a human organism, has 32 chromosomes, how many chromosomes will be in a gamete of that same organism? Would it be A, 8, B, 16, C, 32, or D, 64? So how many chromosomes would be in the gamete? If you said 16, I would agree with you. So basically the muscle cell is a somatic cell. It's a typical body cell. Would be diploid, so that would be 32 chromosomes. So in order for it to be haploid, a gamete, it would be half as much, so that would be 16. All right, so we're done with meiosis 6.6. So by now you should be able to describe the behavior of chromosomes during meiosis, as well as describe cellular events during meiosis. You should be able to explain the differences between meiosis and mitosis and explain the mechanisms within meiosis that generate the genetic variation among the products of meiosis. So in our last video of chapter 6, we're going to look at what happens when we have errors in meiosis and how that results in inherited genetic disorders.